Welcome to another segment of IGS Stand Together. I'm Ben Kreitz. In this episode, we explore the power of genetics and some tips for submitting DNA samples and using genomic-enhanced EPDs to better your herd. Joining me are Molly Diefenbach, American Simmental Association Lead DNA Department, and Dr. Elizabeth Dressler, American Simmental Association Lead Geneticist. Welcome, Molly and Elizabeth.
Hi.
Sure, glad to have you guys here today as we talk through the power of genomics. And Elizabeth, I think a great place to start is, could you maybe share why it's important in today's industry to collect DNA samples?
Sure, yeah. So really, since the advent of genomics, it has sort of become the gold standard within the industry. And I think producers that are making selection using EPDs have really kind of come to expect that is part of the process and part of the prediction of their EPDs. And so it really, the benefits of it kind of can be grouped into sort of two different buckets. One of those would be parent verification. And so even in modern day genetic evaluation, sometimes we get the sire or the dam wrong. And so whether that's bull jumped the fence, we switched our AI straws, or maybe just calves got switched around at birth and we have the wrong dam. And so that does happen, and thankfully we have the genotypes and the DNA to go back and make those corrections and update the pedigree, because that does have an impact on what their EPD will come out to be. And then the other sort of bucket would be getting genomically enhanced EPDs. And those are really, like I mentioned, sort of the gold standard. And when it comes down to it, EPDs are prediction. And so the genotype is an additional source of information that gets incorporated into that EPD. And with that increases the accuracy of those EPDs because we have more information available. And that can be done at a younger age, earlier in life. Genotype can be collected way sooner on in a life compared to maybe, say, gathering up carcass records on progeny. And so with the combination of parent verification, increased accuracy, all of that really leads towards greater genetic gains possible by using the GE-EPDs.
We're adding more confidence to what the true value of those EPDs actually are.
Correct, yes. With that, like it's adding in an additional source of information. Our accuracy will go up, meaning there's sort of less risk associated with that. We're more confident in that prediction with a higher accuracy.
Sure. And Molly, we're fortunate that there's actually multiple ways, right, to submit a DNA sample. And what are some of those options that producers have to utilize?
Yeah, so we get that question a lot. There's three main methods for DNA collection. The first would be hair, and then blood, and then tissue samples, or TSUs. I would say our gold standard today is going to be those TSUs, those tissue sampling units. There's a lower failure rate. There's no age or sex, twin status limitation on that. Hair samples, you want to collect once the calf is after 6 months of age, and you actually cannot submit blood samples on twins. There's some skewed results there. So our gold standard is tissue, but we've had some weird ones too. We've had bulls buried in landslides, and in order to work with their insurance companies, they have to do that parent verification and cut off an ear. We've had ears sent to us. We've had leg bones that have gone to the lab to have DNA extracted. So really, there's a whole variety of methods to collect DNA, but primarily we're looking at blood, tissue, and hair, and we can also do semen straws as well.
Okay. And if a TSU sample is the gold standard today, if a producer collects those at their shoot site and then maybe they have to wait 2 or 3 days or maybe next week until they have them all collected to mail them in, how should those samples be handled? What's the best way?
I would say all DNA samples should just be stored in a cool, dry place. Don't have those TSU samples rolling around on the dash of the pickup truck out in the back pasture. But yeah, a cool, dark place, and they can be stored indefinitely.
And don't even need to be stored in a refrigerator then?
Nope. Once they get to the lab, we handle the sample storage, and they are kept frozen. But as the producer, you do not have to do anything special with those tissue samples.
So, Molly, how early should producers be submitting DNA samples on on their calf crop?
On their calf crop, I would say as early as possible.
So it could be done right after birth?
It could be done at birth. We have a lot of producers. It lines up with their timeline to collect samples at brandings, at weanings, but really as early as possible.
If we think, though, about those genomically enhanced EPDs, Elizabeth, being added into the genetic evaluation, and Molly, from your perspective, how soon in advance do producers need to send in a DNA sample for that to be included into the genetic evaluation?
So the evaluation is run once a week. When the DNA comes back from the lab, that genotype information is shared with our evaluation. And so that would take a full week to run after the sample is done processing.
And there'd be a typical maybe time period that sample would be at the lab. That it's not immediate, right?
Right. I would say, when it's not the busy season for us or the lab, to expect DNA testing to take about 4 to 5 weeks on the safe side, and then an additional week for those genomically enhanced EPDs to show up online.
Sure. So, Elizabeth, if the breeder submits a DNA sample and sends it into Molly's team, and it goes to the lab, how does a producer know that those EPDs are now a GE EPD?
A lot of different breed associations will have that designation for that animal on the animal registry page. And so if you're looking at an animal specifically, I'll just use Simmental as an example. You go to that animal's page on herd book, and it will designate, it will show GE EPD right there on that website next to the animal's name, and you'll know that that is a GE EPD, and the genomics are incorporated in the prediction of that.
Sure. And you talked a little bit about it earlier in our conversation, but could you share again some of the benefits of having genomically enhanced EPDs?
Yeah, so with genomically enhanced EPDs, it really has allowed the beef industry to make greater and faster genetic progress, just like we talked about earlier a little bit with increases in accuracy. And so with increases in accuracy, it's another source of information going into that prediction, and so that leads to greater genetic progress, and you're able to make that you're able to collect that sample earlier in life and make those selection decisions before you keep that animal around for too long.
So, Molly, in addition to genomically enhanced EPDs, what other information can be determined from a DNA sample?
There's a whole host of results that we can get from a DNA sample. Everything from coat color, horn polled results, parent verification is a big one that Elizabeth touched on earlier, just verifying that pedigree. We also, Simmental specific genetic defects as well, tracking that in your herd, getting your bulls and your dams cleared of those defects is really important moving forward. Red Charlie is another one that's specific to Simmental as well.
Something as easy as just a TSU sample at birth provides us all of this information that otherwise we really wouldn't know what the true answer is.
Absolutely.
Then you both fortunate to visit with a lot of producers and members in both of your roles. So what are some of the common questions that you receive from breeders regarding DNA samples and regarding genomically enhanced EPDs?
One thing I'll mention maybe is that it's important, if you can, to collect those DNA samples on an entire contemporary group. So for example, your entire calf crop. At the Simmental Association, we do have a couple of different programs or DNA programs that are available for members to collect that DNA sample on the entire contemporary group. Whether that's the calf crop, you would go maybe towards the CCG or the calf crop genomics program. If you're wanting to get that information on your entire cow herd, we have CHR or cow herd roundup. And so we really encourage people to collect it on all of your animals, not just a select few. And we do have those programs available to help members out from a cost standpoint as well.
Molly, from your perspective, I know you field a lot of phone calls as breeders, as maybe a bull sale is approaching and there's questions or maybe something that's pending. Yep. What are some questions that you hear from breeders that reach out?
A lot of time it's timeline, just how long is this going to take, trying to work it in, especially this time of year with catalog deadlines and bull sales. And again, try to plan for 4 to 5 weeks on that. It's not instantaneous. You don't get results back necessarily that same week. So that's a big one, just understanding the process and the timeline, helping members with Simmental specific rules and bylaws and what testing is required for that, beyond the genomically enhanced EPDs. And fortunately, our programs are set up that if you are, AI sires, for example, donor dams, walking bulls, they have to have specific testing done. They have to be cleared for certain defects. And just understanding those rules and talking with producers and making sure that they're able to register their animals and meet those DNA requirements as well.
So the sooner the better, that a producer is able to collect a DNA sample and submit it would be a great piece of, or a great recommendation.
Yes.
Well, Elizabeth and Molly, thank you for sharing these helpful reminders as we consider important timing on the ranch. And for more educational segments like this one, please visit internationalgeneticsolutions.com.